Alrighty, getting started here for week 10. We're going to be starting off with chapter 12, so that's where we're going to start looking at the origins of life and diversity. So first of all, what is life? First of all, I also want to point out that I finally said the thing that this right here, what is life? It's like the name of the whole book. So we're finally doing that. It's happening. Life is used to describe two things. So when we're describing a living thing, we're describing something that is able to replicate or reproduce, and then it has to have some kind of metabolic activity. So it has to be acquiring and using some kind of chemical energy. And that's pretty vague, but enzymatic activity falls under that. Things like photosynthesis and cellular respiration, which we talked about, fermentation, all those things count as metabolic activity, or where we're breaking and then reforming chemical bonds. This gets a little tricky because some things act in a lot of the same ways that living things do, but aren't quite considered living by many scientists. So some things can replicate but not do metabolism, like viruses. Uh, so viruses still undergo evolution. They still have genetic code. It can be in either RNA or DNA, depending on the virus. But what happens is because they still go through some form of reproduction and they're self-replicating, and sometimes there's mistakes along the way, just like for normal cellular reproduction, they evolve. Their mistakes are made. Sometimes mistakes are favorable in biology. So the difference here is that because these viruses don't have any of their own cellular machinery, they have to hijack other cells. So their metabolism is really facilitated by the metabolism of other creatures. Other creatures essentially become virus factories. Other cells will become virus factories for the virus that infects them. So the virus itself isn't doing any of the metabolism. It's not doing any of the work here. It's all hijacked proteins from the host cell. So the question uh, is it living? Is it non-living? Is hijacking other cells' metabolisms enough to qualify it as living? That is a difficult question, and I don't want to lean too heavily on my opinion here, because I want you guys to write to me with your own opinion. So going off of what we just talked about on the bottom, we have a virus, a rhinovirus to the left down here, and we have an amoeba down here to the right. Which of those do you think is living or non-living, and why? Um, where will you draw the line? And then going along with that, a little bit of a puzzle. At the top, which of the things in this image are living? Which of the things in this image are living? So we have a lot of different things there. We have a river, we have some rocks, we have a campfire, mountains in the background. I want you guys to list off all the things that you consider living in that image. All right. And with that, we will move on. All right. Biodiversity. So, the diversity of living things is what we consider the biodiversity. So the current biodiversity of the Earth is all the things that are living on it today. The combination of all the genes, all the organisms, all the ecosystems that have been put together by these organisms. All of that is diversity. Is the biodiversity, the diversity of life. So bio means life, diversity. We're just using it as diversity here. So the diversity of life, biodiversity. We can refer to this in past, though, or in future. So if we're worried about the future biodiversity because of climate change, that is a proper use of the term. Also, if you're looking at the biodiversity of the past, you could say the biodiversity during the time of the dinosaurs. So that this is a sort of just a general term for where the current range of genes and organisms is on the world right now, what they are. All right. Let's look into the origins of Earth here really quick. So the origins of Earth, we believe, started with essentially hydrothermal vents. So early Earth, before life, there was a lot of chemicals sort of just floating around. We did still have oceans, we had mountains, we had volcanoes, we had volcanic activity, we had all these things. And in pools in early life, the sort of building blocks for organic molecules began to arose from the chemical soup of the time. And we can sort of replicate this in experiment. So if you take a flask, you fill it with hydrogen gas, methane, which is naturally occurring, and ammonia, NH3. You sort of stream it past you just stream it past a light, essentially, and constantly rotate it through a heater. There's like essentially there's separate condensation and dripping happening here, and through the separation of these chemicals and just the application of heat and light and cool, we have a lot of these chemicals forming amino acids. So we have 5 different amino acids detected in this experiment. So remember, amino acids are those building blocks for proteins. So we have, we've already simulated the formation of amino acids just with inorganic materials. And it's not impossible to think that amongst these particles, some of them would eventually be able to self-replicate. They will just cause more of their own kind to be made. And as soon as you have that self-replication happening, you have sort of the spark that creates life, essentially. As long as you have a stable system where you have something creating copies of itself, you will eventually get these early life forms. And if we left an experiment like this running long enough, or with enough diversity of materials as well, given a few million years, we might see early life forms. Obviously, that's beyond the scope of an experiment, so we sort of have to simulate out further than that and make sort of logical leaps as to what we would see. But yeah, phase 2, here we have the formation of these self-replicating molecules. So things like RNA. RNA can have some metabolic processes like tRNA, which helps bring the amino acids in during translation. tRNA has some enzymatic properties to it. So some theories say that RNA was the first precursor to cellular life, the very first thing that we might start to consider living. This is also a place where that virus idea gets a little confused. Is where do we call the origin of life? If we think RNA molecules that were self-replicating were the first types of life, then where do we draw the line with current molecules of RNA that put themselves into a protein coat and then infect other cells to make them make more protein coats and RNA strands for themselves? So it's a complicated issue, to be sure there. But yeah, we believe life started off as these self-replicating molecules, which eventually were likely partly at least RNA. And we have some evidence of the earliest forms of life. There is some fossilized microbes that we found. So based on the carbon dating of the rock, we were able to put it about, or not carbon dating specifically, but radioisotope dating. They were able to put it at 3.4 billion years old. for the uh, for the fossil. So the development. So after we get self-replicating molecules, we'll have membranes developing as the next sort of leap in evolution that we're going to be discussing. So we have these RNA molecules just floating around in some kind of primordial ooze, and eventually some of them develop the trick to start coating themselves in a semi-permeable membrane. Um, Lipids were probably present at the time, so one of the molecules would organize the lipids around it to protect it. At least that's a logical guess we can make, because it is a very favorable trait to have developed, to be sure. Most of our organisms we see today, most things on Earth today, have a cell membrane. Most living things have a cell membrane. So, of one form or another. But anyways, we have membranes will enable these organisms also to do different things, like different kinds of metabolism are only possible if you have a membrane-bound organelle, or just a membrane in general. Being able to keep concentrations of different molecules close to you enables you to do things that weren't previously possible. So those are the three main steps that we believe is the accumulation of organic molecule precursors, so things like ammonia, hydrogen gas, methane, the formation of self-replication, things like RNA that can make copies of themselves, and then eventually the development of a membrane. But yeah, so that is our brief look into the origin of life. Next up, let's look at species. What makes species distinct from one another? Where do we separate out species? It's important to remember that we can't just go based on looks, similarities, because um, a lot of times the phenotypes are hard for just the human senses to distinguish, especially an untrained eye. So while two human beings here, we have Cristiano Ronaldo and of Real Madrid and Kerry Washington. They're both members of the same species. We're all humans, but then we have other things that look fairly similar to one another, but are definitely different species. So we have two different flowers here, the Persian buttercup and the pink rose, and we have a orangutan and a tamarin next to each other in the other pictures. So important to realize that just because they look similar does not mean that they are the same species. Specifically, when we're describing species, we are talking about two organisms. If you're a member of the same species, then you can reproduce and have offspring that are capable of having offspring of their own. They're not infertile children. An example of infertile children would be a horse and a zebra can have a child, but it is infertile. A horse and a donkey can have an offspring, but it is infertile. It is a mule. Lions and tigers can have offspring. They're still close enough to have offspring, but they are the offspring are all infertile. The ligers are infertile. So some organisms are close enough to where they can still have offspring, but those offspring will not have children of their own. So that's a sort of dead end, genetically speaking. So when groups of organisms become genetically different enough, they can no longer reproduce offspring with one another, and then that is when we consider species to be reproductively isolated. So when you can no longer have offsprings with members of a similar group, you are reproductively isolated. When that occurs, the two groups are considered separate species. So this is one way of looking at species model. All right, and to the left, we got some alpacas. I've heard of guanacos. I don't. They look like alpacas. Interesting. Anyways, and we also have a picture of that horse zebra hybrid we mentioned earlier. So they'll create a child. That child is healthy enough, except it is also infertile, so it cannot have offspring of its own. So how do we name species? So I might have mentioned it earlier, but humans are Homo sapiens. So going along with that, our genus is the Homo genus, and our species is sapiens. There's been other groups from the Homo genus before us, but have since gone extinct. Most recently, Homo neanderthalis, which is the Neanderthals. The naming scheme dates back to Carolus Linnaeus in the mid 1700s, and it shows its age in a lot of ways that if you study it in greater detail, you'll see, like the fact that he had these sort of linear breaks in diversity in organisms, but the truth is that the web of life is a mess. The tree of life is a mess, and it's impossible to just have distinct levels past a certain point. But we can still do our best here. So we do have domain, phylum, or domain, kingdom, phylum, class, order, family, genus, species, the classical Linnaean taxonomies. But within these, scientists have had to add subspecies or subgenuses or subfamilies because organisms don't break out perfectly within 4 categories or within 7 categories. But yeah, let's look at this zebra here. So the zebra is part of eukaryotes. It's a eukaryotic domained organism. It's in the kingdom of Animalia. So out of eukaryotes, there is Protista, Plantae, fungi, and animals. We have its phylum is Chordata. That's things with a spinal cord. Class, mammals, so organisms which give birth to live young and use mammary glands. That's where the mammalia comes from, the mammary glands to feed those young. And now we're getting to where I have less knowledge of. So Perissodactyly, then Equidae, Equus, and then the species name, or specifically, is Equus quagga. I'm making total educated guesses at those pronunciations, by the way. At least some of these pronunciations. That's just sort of how it is in science sometimes. People pronounce things weird or differently. But yeah. So the highest order is domain at the top here, is where we can bounce back to here. And yeah, these categories work for a wide variety of usages, but just keep in mind that sometimes we have to add in addendums here and there where life kind of threw us a curveball and has different differentiations than we had originally planned when we had classed them out. But yeah, species don't always work in the classical definition well. So biological species concept is going back to this idea that we have reproductive isolation as the key feature for separating out species. They're unfortunately not always so easily defined because we have things like asexual species. So either bacteria, or if you consider viruses living, viruses as well, where they reproduce asexually. A lot of larger organisms also reproduce asexually. So if they aren't going through sexual reproduction, how do we consider them sexually isolated? We really can't. So how do we define species then? that gets a lot more tricky. That's where we have to look more at the genetics that are different between them. But how about if we don't have any of the genetics? When we look at fossils, we don't have any genetics. So classifying fossil species is another interesting example of when it's very hard to sort of uh, dissimulate different species. How do we isolate one species when we don't have any genetics and all we have is morphological figures? We have their skeletons, their bones, their hard structures, sometimes feathers. If we're very lucky. Um, but usually it's very dense structures. So where do we draw the line between two specimens? If like, do we consider them to be the same species? If we only have a partial specimen, it gets even more difficult. So these lines can be a little bit hard to draw. Another thing is that there's almost never a specific moment when two groups of organisms become so different that they are now a species. Usually this happens over the span of a gradient. There's like a gradient from one side to another where like maybe just geographically isolated organisms start to become very different but are still members of the same species. A lot of times you'll have like maybe a mountain dwelling organism, a hill dwelling organisms, and then a plain dwelling organisms. The two that are in closer habitats can still breed. The two that are in closer habitats on this extreme can still breed, but if you try to breed across, then they cannot have fertile children. So you have a lot of these weird sort of hybrid speciation events where they're not quite species separated yet. They're still very close. Also, let's see, classifying. So two non-interbreeding populations may be connected to each other through gene flow. So we could have genes being spread across that gap, like I was saying earlier. But there's no exact moment, there's no exact separation between the species. Like this one isn't exactly distinct from this one because like their children's children could still have offspring together. So it gets really weird there. Um, it's just biology not playing by our rules, but we're still doing our best to define and classify and learn more about them. So it's just really hard to classify those hybrid species a lot of times, and they are quite common. Um, this is a pretty natural process, especially in uh, when we're looking at current times. Like every time a new organism is possibly speciating from one another, um, we have to consider where we define that species barrier. All right, so that is the end of today's slides. With that in mind, our goals for next class is 12.11 through 12.18. That's pages 397 through 407. And we'll be continuing our slides right around 12.7. So we got through about 12.7 today on the slide side. We'll be breezing through the rest of that on Wednesday. And with that, I hope you guys all have a great day. Goodbye.